Biology, Part 176. STEM functions. STEM functions. The most important functions of the STEM are support, transport, and growth. In a stem consisting of primary tissues, the function of support is shared between several different types of cells. The living cells of the epidermis, cortex, and pith all take in water by osmosis. As a result, individual cells develop an internal hydrostatic pressure called turgor pressure, which causes protoplast to push outwards against the cell wall. This is explained in the unit 33. The turgid cells are rigid and resistant to bending. If the cell lose water, they become limp or flaccid, and the stem wilts. The collenchyma and the sclerenchyma cells and the heavily lignified cells, xylem tissues, also function in support. In most terrestrial planes, the major mechanical stresses are imposed by the wind, so that the stem must be able to resist bending to remain upright. The vascular bundles containing the xylem are tough and inextensible and perform the same function as steel rods in the reinforced concrete. Their arrangement as rings within the stem provides very effective resistance to wind stress. In the stems of some plants, for example in sunflower, the helianthus, the vascular bundles are strengthened by additional sclerenchyma fibers, which form a bundle cap. The sieve tube elements and companion cells of the phloem and the tracheids of the vessel elements of system are specialized for transport. The mechanism involved in their functioning are complex and important and will be discussed in our tutorial. As already explained, that the primary growth in stem takes place by differentiation from the apical meristome and three primary meristemic tissues. In addition to these essential functions, some stems are specialized to perform other tasks. Photosynthesis and the storage of food water are among the most common. In cacti, for instance, the stem is capable of storing large volumes of water, and the leaves are reduced to spines so that the stem becomes the main organ of photosynthesis. Combs, stems, tubers, and some types of rhizomes are modified stems, which act as food storage organs and can be used as the means of asexual reproduction. Most spines and thorns are modified stem branches. Secondary growth. Primary growth is responsible for the increases in stem length, increases in stem diameter, and the continued development of the plants from year to year requires a different type of growth called secondary growth. Plants with an annual life cycle like that of the broad bean usually have a limited capacity for secondary growth. The adult plants simply wither and die as winter approaches, leaving seeds which will germinate in the spring. On the other hand, the perennial plants, for example trees, rely on secondary growth to reach a large size and may survive for many years, producing seeds in a number of successive seasons. The main stages of the secondary growth are outlined in our figure. When vascular bundles are first formed, not all the procymbal cells differentiate into the primary phloem or xylem. In the early stages of secondary growth, the remaining cells become active once more, forming the regions of fascicular cambium inside the bundles. These produces new phloem cells called the secondary phloems to the outside, and the new xylem cells called the secondary xylem towards the inside, of course. At about the same time, some of the parenchyma cells in the pith race between the bundles are stimulated to divide and start to form a linking region known as interfascicular cambium.